The nutritional improvement and quality improvement by employing transgenic technology will be discussed in this lecture. So, biofortification is a process of breeding food crops that are rich in bioavailable micronutrients. These crops biofortify themselves by loading higher level of minerals and vitamins in the seeds and roots, which are then harvested and eaten. Biofortified crop varieties could naturally reduce nutrition-related health problems in hundreds of millions of people. So, when we talk about strategies to address malnutrition, there are four different strategies to address the malnutrition problem. The first one is diversification of food. The main issue here is diverse diets are often costly and not locally available, and the main challenges are like limited market access to a variety of foods, cultural or seasonal dietary habits, and cost barrier for animal source foods, fruits, and vegetables. So the second one strategy is fortified foods. The issue with the fortified foods are like commercially fortified foods are not affordable or accessible in poor communities. The main challenges are fortified products such as enriched cereals, fortified oils often require centralized processing and purchasing power. So third strategy is medical interventions. So the issues here expensive and require sustainable healthcare systems, and challenges includes costs and supplements and clinic visits, and inconsistent access to trained health workers or supply chains. So the fourth one is actually developing nutrient-rich crop varieties. So this could be a kind of a potential game changer for resource poor settings. So this is a cost-effective, sustainable strategy that deserves emphasis. And main advantages are here like one introduced, seeds can be planted each season, grown and consumed locally, no additional cost for purchase or transport, and increase both food security and nutritional value. So when we discuss about merits of biofertilization strategies, so there are three different options are available: mineral fertilization or conventional breeding and metagenesis, the known as transgenic approaches. When we talk about mineral fertilization, it's a simple, inexpensive, rapid enhancement, but it only works with minerals, not suitable for iron, very dependent on crop and cultivars, not possible to target edible organs. So when we discuss about conventional breeding and metagenesis, it uses intrinsic properties of crop, few regulatory constraints, but it depends on existing gene pool only. It takes a long time to breed the particular trait. Traits might be need to be introduced from wild relatives. That's again one big issue, and possible intellectual property constraints. So when we focus on the transgenic approaches, it's kind of from rapid, unconstrained by gene pool, and targeted expression in edible organs. That's one very beautiful about this particular technology and applicable directly to light cultivars. But the main issues here is regulatory landscape, political and socioeconomic issues relevant to transgenic plants, and sometimes possible intellectual property constraints also. So vitamin A deficiency mainly affects preschool children and pregnant women in the world. Vitamin A deficiency causes blindness in children. The cure for this is to provide vitamin A supplements as capsules. An alternative and viable approach to alleviate this problem is to provide beta carotene, that is actually the provitamin A in the grains, such as in wheat and rice. So human can synthesize vitamin A provided with precursor molecule beta carotene, also known as provitamin A. Beta carotene is assumed to be converted to retinol in the animal gut. Milled rice is normally devoid of beta carotene and other colored phytochemicals. The rice plant can naturally produce beta carotene in its leaves where it is involved in photosynthesis. The beta carotene is an accessory pigment that is essential for photosynthesis. It is synthesized from the isoprenoid pathway in all green tissues. Carotenoids are fatty carbon compounds produced from the precursor molecule via a biochemical pathway localized in plastic. The fatty carbon backbone of beta carotene is phytone, which is assembled by combination of two 20 carbon general diphosphate, that is often called as GGPP molecule, by the enzyme phytone synthase. Double bonds are then added to phytone through a series of desaturation steps to produce lycopene, an antioxidant compound found in most plants that contribute to the red color, particularly in the vegetables like tomatoes. Finally, lycopene can be converted to the beta carotene by the enzyme lycopene cyclase. Plant does not normally produce the pigment in the endosperm where photosynthesis does not occur. Although rice seeds does not contain beta carotene, a precursor, that is GGPP, is produced. GGPP is not converted further into beta carotene as it lacks enzyme phytone synthase, phytone desaturase, and beta carotene desaturases. So Dr. Enko Potrikes in Switzerland and Peter Bayer in Germany are the two persons responsible for transforming rice with two additional genes to produce beta-carotene rice endosperm. So to synthesize beta-carotene rice seed, the transgenic rice plants were generated that express two additional genes in the endosperm of rice seed. Golden rice was created by transforming rice with only two beta-carotene biosynthesis genes. First gene that encodes for phytosynthase, obtained from daffodil plant, was expressed using rice endosperm specific glutenin promoter GT1. Actually, this shows tissue specific expression expressed only in the endosperm for the production of phytone in rice grains. Because the daffodil gene products are normally found in plastids, they already contain sequence for plastid transit peptide to direct the newly synthesized photosynthesis enzymes to plastids. So the second gene CTR1 carotene desaturase genes obtained from the bacterium Erwinia rudibora was expressed under the cauliflower mosaic virus 35S promoter. This enzyme adds double bonds to phytone to produce lycopene. This bacterial enzyme does the function of two plant enzymes via, that is phytone desaturases and beta carotene desaturases. This bacterial gene encoding desaturase was modified by addition of transit peptide to direct it to plastids following translation. Finally, lycopene is converted into rice grains by an endogenous lycopene cyclase to beta carotene, that is an yellow product. Transgenic rice produced golden colored seed that accumulated up to 1.6 microgram of total carotenoids per gram dry weight, which is about 50% of this found as beta carotene, that is actually zero point 0.8 microgram of beta carotene per gram. So the further work saw the development of golden rice 2, in which the daffodil phytone synthase gene was replaced with its more efficient maize phytone synthase PSY gene along with the Erwinia rivera carotene desaturase CTR1 gene expressed under a rice glutenin specific promoter GT1. Transgenic rice yielded up to 37 microgram of total carotenoids per gram, preferentially accumulated beta carotene up to 31 microgram per gram, thus produced 23-fold increase in carotenoids compared to the first generation of golden rice. It is estimated that by eating modest amount of golden rice too, enough beta carotene can be provided to overcome vitamin A deficiency. That is remarkable work by using the transgenic technology and to improve the nutritional requirement of one particular trait. So this is appearance of golden rice, which is rich in beta carotene, that is precursor of vitamin A. And the Philippines is the first country to approve GMO rice to fight malnutrition. This is for information. So next one, we'll discuss about vitamin E metabolic engineering. Vitamin E is important for human and animal health. The daily requirements for vitamin E in men and women have been increased to 15 to 30 milligram. The primary source of dietary vitamin E comes from plants. Vitamin E metabolic engineering in plants is aimed at improving the vitamin E content and regulating composition of vitamin E. Tocotrienols are the major form of vitamin E in the seeds of most of the monocots and limited number of dicots also. Among these derivatives, alpha tocopherol has the highest biological activity for human health. That means the goal of vitamin E biofortification is to somehow to increase the contents of vitamin E. That is actually convert all the other type into alpha tocopherol. So again here, as like in the case of golden rice or vitamin A synthesis, general diphosphate synthase catalyze the formation of 20 carbon general diphosphate, which is the universal precursor for carotenoids, as we discussed earlier, and terpenes, gibberellins, and vitamin E. So GGP reductase and gamma methyl tocopherol transferase are two enzymes responsible for tocopherol biosynthesis in plants. Of the four naturally occurring species of tocopherols, vitamin C's, alpha tocopherol is the most important one to human health as it has higher antioxidant ability than other forms of tocopherol. So vitamin E is a group of eight hydrophobic compounds, also known as vitamins, and as said earlier, the most potent of which is alpha tocopherol. The Sikimate pathway generates homogenesic acid, which forms aromatic ring of tocopherol, whereas the side chain is derived from phytyl diphosphate, a product of MEP pathway. So these precursors are joined together by HPT to form the intermediate MPBQ. The MPBQ is the substrate for two enzymes, tocopherol cyclase and MPBQ methyl transferase. Tocopherol cyclase produce sigma tocopherol. Both gamma tocopherol and sigma tocopherol are the substrate for gamma TNT, that is actually producing alpha and beta tocopherol respectively. So the levels of vitamin E activity can be increased by increasing the total amount of vitamin E, or the another way what we can do is shifting the metabolic flux towards alpha tocopherol. So in 1989 itself, arabidopsis genes encoding gamma TMT in arabidopsis seeds actually overexpressed, and that actually resulted in fundamental shift from gamma or sigma to alpha and beta tocopherol. The expression of arabidopsis homogenesic acid pronayl transferase produce twice the level of vitamin E found in normal seeds. Transgenic soybean expressing Arabidopsis MPBQ methyl transferase and gamma TMT. So significant elevation in total amount of vitamin E activity and an eightfold increase in the level of tocopherol from its normal 10% of total vitamin E to over 95%. So the next part of this lecture Uh, we're going to discuss about quality improvement through transgenic approach. So the quality here, the manipulation of shelf life in fruits and vegetables. So fruit ripening is characterized by ethylene production, fruit softening, change of color and flavor. So ethylene triggers the ripening in climatic fruit. Softening is due to cell wall degradation, that is polyelectronics PG and pectin methyl esterase. So, fruit ripening is a complex, genetically programmed developmental process involving numerous physiological and biochemical and structural changes that transform the fruit from an unripe, green or hard state to ripe, soft and colorful and flavorful state. So, this process is tightly regulated by hormones, particularly ethylene, which is crucial in climatic fruits like tomato. So here is a breakdown of major developmental changes during fruit ripening process. The first one is actually the color change. So often we can call it as a pigment transformation. So chlorophyll degradation takes place here. The green pigment in the fruit breaks down. Carotenoid accumulations, especially lycopene, red pigment, and beta carotene, orange, leading to the red or orange appearance, and controlled by phyton synthase and other carotenoid biosynthetic enzymes. And the second one is actually sugar accumulation and acid decline, taste development. So glucose and fructose levels rise, improving sweetness, and reduced organic acids, especially citric acid and malic acid, leading to a sweeter taste balance. Changes due to enhanced sugar transport and altered metabolic activity inverted sugar transporters. And third one is actually the ethylene production, hormonal regulation. So tomato is a climatic fruit, meaning ripening is driven by a spike in ethylene production. Ethylene triggers ripening-related gene expression, including those for softening enzymes, color-related enzymes, flavor biosynthesis enzymes. Then fourth process is fruit softening, textural changes. Cell wall modification, degradation of pectin and cellulose by enzymes like polyelectrolysis (PG), often called as PG, pectin methyl esterases (PME), and expansions and leads to loss of firmness, making the fruit soft and eatable. And fifth one is respiration burst, climatic rise. Increased respiration rate, CO2 release marks the onset of ripening, fuels the synthesis of ripening related compounds and energy demanding process. And sixth one is flavor and aroma development, formation of volatile organic compounds that gives ripe fruits their aroma, derived from lipid, amino acids, and sugar metabolisms. Key aroma compounds include hexanols, cis-3 hexanols, 6 methyl 5 heptane 2 1. And the seventh one is changes in synthetic metabolites, accumulation of phenolics, flavonoids, and antioxidants like lycopene. These compounds contribute to color, nutrition, and health benefits. And the final one is gene expression reprogramming, massive reprogramming of gene expression. Ripening is regulated by transcription factors like RIN, NOR, CNR. So climatic fruits shows dramatic increase in the rate of respiration during ripening and will respond to ethylene for ripening. So examples are apple, banana, mango, and tomato. And non-climatic fruits do not show dramatic increase in the rate of respiration during ripening and do not respond to ethylene for ripening, such as citrus, grapes, pineapple, and watermelon. So the transcription control of fruit ripening and conserved ripening regulators. Although considerable efforts has focused on ethylene synthesis and response, only recently have inroads been made into understanding the ripening control before ethylene, a regulatory system that is possibly conserved between climatic and non-climatic species. Recently, the mutated genes underlying the ripening mutants RIN and CNR were cloned, providing key insights into the regulation of ripening process upstream of ethylene synthesis. The RIN and CNR mutations are recessive and dominant mutations respectively, which block the ripening process and result in fruit that are unable to uh, respond to the ripening associated ethylene. So the ring mutation is especially interesting as it has been bred into many commercial tomato varieties to slow ripening and extend the shelf life. Rin encodes Madbox G proteins of spectral while CNR is an epigenetic mutation that alters the promoter of methylation of squamous promoter binding protein. Both these transcription factors are required for ripening associated increase in respiration and ethylene characteristics of tomato and other climatic species. So another tomato transcription factor that is LEHB-1 has also recently been characterized and shown to play a central role in fruit ripening process. LEHB1 encodes a putative HD-GIP homeobox protein that binds to the promoter of ACC oxidase, which encodes a key enzyme in ethylene biosynthetic pathway. Putative LEHB1 binding sites are also present in a number of other ripening-related genes, including LEACO2, LEPG1, and LEMAX-RIN. And so it is possible that LEHB1 might also regulate these ripening-related genes directly. These are actually the phenotype of different mutants in tomato. So this is the wild type, and these are the mutants, and you could able to see very clearly that the ripening and other related processes are delayed in the mutants compared to the wild type cultivar. Next one is that cell wall changes. It is rich in polysaccharides, are degraded and solubilized during ripening process. Loss of neutral sugars such as galactose and arabinose. Enzyme responsible for cell wall hydrolysis are PE, PG, and PL, beta galactidase, and cellulase. So in fruit, the cell walls are mainly composed of polysaccharides, including pectin. During ripening, a lot of this pectin is converted from a water insoluble form to soluble one. The fruit will become less firm, and as the structure of the fruit is degraded. So delaying fruit softening is one of the target for breeding, and respiration is the process by which stored organic material, carbohydrates, proteins, and fats are broken down into simple and products with release of energy. Respiration involves degradation of food reserves. So this is the ethylene biosynthetic pathway. So and methionine recycling in plants. Ethylene is synthesized in two-step pathway that involves the enzymes AC synthase and ACC oxidase. Yes, adenosyl methionine is continuously replaced by a methionine cycle, although it can also be diverted into the biosynthesis of the polyamines, spermidine and spermine. Nitric oxide inhibits the activities of ACC synthase and ACC oxidase, and it also affects the SAM synthetase by nitrosylase event. So if the target is ethylene biosynthetic pathway to delay the fruit ripening means blocking the expression of genes induced by ethylene could be one approach, and blocking ethylene synthesis is another approach. Blocking the perception of ethylene is another one approach. So downregulation of ACC synthase or ACC oxidase. So by using antisense RNA technology, that could be one strategy, and the expression of ACC deaminase from bacteria that actually metabolize ACC, resulting in a less ACC available for ethylene production. This best example is in tomato. And expression of SAM hydrolase. SAM is converted into homoserine, example in melon and tomato. Or the another one approach is downregulation of polyelectronics, antisense, RNA technology, RNA technology. So improving fruit shelf life by inhibiting the expression of ethylene biosynthetic genes by antisense technology or RNA technology. So ethylene is synthesized in higher plants from two key enzymes, that is ACC synthase and ACC oxidase. In tomato, ACC synthase gene active during ripening was silenced using the antisense techniques, effectively reducing the production of ethylene by the ripening fruit by 99.5%. While the control fruits begin to produce ethylene around 50 days after the pollination and immediately undergo a respiratory burst, genetically modified tomatoes produce minimal level of ethylene and fail to produce the respiratory burst, thereby improve the fruit shelf life. So the examples are in banana as well as in tomato. The shelf life of tomato fruits increased by inhibiting the expression of ethylene biosynthesis genes by antisense technology or RNA technology. So another one target is pectate lyase as an effective target for fruit shelf life improvement in tomato. So both RNA technology and CRISPR-Cas9 based technology were used. So in the case of RNA techniques, reduced PL activity, delayed softening, and without impacting other aspects of ripening. So in the CRISPR-based genome techniques, three genes were targeted: PL, PG2A, and TPG4. And among these three, only PL resulted in farmer fruits. This is the wild type and mutant phenotype. It also shows that targeting pectate lyase as an effective target for fruit shelf life improvement in tomato.